Let me show a very small video on protein synthesis.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember,
So you can see here, guanine pairs with cytosine, but adenine will always pair with uracil because it is an mRNA strand. So adenine will always pair with uracil when there is synthesis of mRNA in the DNA, in the nucleus. So RNA uses uracil instead of thymine. Okay.
if the DNA strand looked like this, the complementary mRNA strand would be the
So what will be the complementary mRNA strand for this? A, adenine pairs with thymine in DNA, but in mRNA it will be uracil. Thymine के साथ आएगा adenine, C, G, G, C. Only instead of A you will have uracil instead of thymine.
complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it formed. It moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA. And that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome, and a start codon must be read. It's usually AUG, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in.
So again, the next tRNA will come, join to the next 3 codes on the mRNA, will supply another amino acid. Okay, so एक अमाइनो एसिड और एक आ गया, तो ये जो पहला वाला tRNA है वो निकल जाएगा और ये अमाइनो एसिड दूसरे वाले अमाइनो एसिड के साथ जॉइंट हो जाएगा. दैट इज़ हाउ अमाइनो एसिड्स गेट जॉइंट टुगेदर विद द हेल्प ऑफ राइबोसोम.
The protein grows until a stop codon is reached. Once the stop codon is read, now we can figure out the sequence of amino acids using the
Now remember, particular nucleotide sequence codes for particular amino acid. Okay, कोई भी random nucleotide sequence कोई भी random amino acid के लिए code नहीं करता है. Only particular nucleotide sequence will code for particular amino acid. So if you see here, we will go through it once. See, I am highlighting this for you. Proteins are synthesized by DNA through RNA. This is called central dogma as per the sequence of nucleotides on DNA. Okay, this process is called as transcription. Next, अब जो mRNA बन गया है न्यूक्लियस में, that will come into the cytoplasm. Okay, what will happen in the cytoplasm? The ribosome will attach itself and it will go on reading the sequence. Now remember, 3 nucleotides will always code for 1 amino acid. So if you look here, this is your mRNA 1, 2, 3, UAG. Sorry. This 3 will code for 1 amino acid. Then another 3 will code for another amino acid. Okay, this is called as triplet codon. This triplet codon was given by scientist Har Govind Khurana. Okay, so three amino acids in a mRNA sequence will code for 3 nucleotides will code for 1 amino acid, and this is called as triplet codon. So ये triplet codon है, ये read होगा by the ribosome and a tRNA will come which will have complementary sequence. Okay, U read करेगा, U के opposite क्या आना चाहिए? A. Okay, then thymine के जगह पे U, G, C. So the tRNA will have a 3 nucleotide sequence which is called as anticodon. The mRNA has codons, the tRNA has anticodon, मतलब इसका opposite. So mRNA codon, tRNA anticodon. This binds together and an amino acid is released. ऐसे ही कर करके tRNA आएगा and the ribosomal RNA will go on joining the amino acids to form a huge protein. This is the process of translocation. Okay. See the next line. The code for each amino acid is of 3 nucleotides. It is called as triplet codon. He gave, Dr. Har Govind Khurana gave triplet codon phenomenon. Okay, 20 amino acids are naturally known to the human body. Okay, he discovered the work of triplet codon and he identified 20 amino acids that are naturally present. Look at the next part. We have already discussed also. We will just go through. Each mRNA is made up of thousands of triplet codon. As per the message of mRNA, amino acids will be supplied by the tRNA. Okay, the tRNA will have anticodon. The mRNA has codon. This is called as translation. फिर जो अमाइनो एसिड आएगा tRNA से, that will be all joined together by peptide bonds with the help of ribosome, ribosomal RNA. So this is called as translocation. So many of these proteins will come together. Okay, many proteins are formed inside the body. These proteins control different functions of the human body. Okay, now remember, just like human beings grow, okay, they produce new offsprings. The characters are also transferred from one generation to another. The genes are also transferred. When characters are transferred, obviously the transfer is taking place due to the help of genes. So many of those genes are transferred from one generation to another without any changes, but sometimes some changes may take place. These sudden changes is called as mutation. One nucleotide or many nucleotide may change their place, and that is called as mutation. The mutation could be useful or could be harmful. Okay, some mutations are so minor कि people will not notice any change. कुछ तो हो गया छोटा सा पर फ़र्क नहीं पता चल रहा, but there are some mutations that can cause various harmful diseases. For example, sickle cell anemia. Okay, so that is why study of mutation is important. If you can see here, see the sequence here, TGC is replaced by GAT. So as we know, three nucleotide will code for one amino acid. अब जो TGC के लिए कोड कर रहा था, the TGC which it coded for some amino acid, but now it is replaced by GAT, so some other amino acid will be produced. Okay, so it changes the function of the body. It could be useful or it could be harmful. It is called as mutation. See, any nucleotide of the gene changes its location, position, that causes a minor change, which is nothing but the mutation. Okay, this is the first part of the chapter, that is protein synthesis.